Hello children, welcome back to my talks, and here I am with standard 11th biology. Okay, so this topic ATP synthesis through chemiosmosis. Okay, so as earlier whatever we have done, if you remember that, I had already told you, okay, that you must be thorough with the other topics which were there. Okay, cyclic and non-cyclic photophosphorylation, splitting of water, ETS chain. Okay, you should be thorough with all that, and hence you will understand this ATP synthesis. Okay, so again, before we begin, if you have not, children, yes, 11th standard biology, I do put these videos on the channel, but then only because you have not subscribed, you don't get it, you don't get the notification. So do subscribe it, children, okay, so that you get the notification, you can see it, you can see it twice, thrice, because as I told you, this is a very important chapter. Okay, from your examination point of view, as well as from you can say medical NEET point of view, because in NEET you get questions from 11th as well as from 12th. Okay, so today what I have decided is I will explain it to you thoroughly from the textbook. So first we come to this, or first should we do with the book? Okay, first I'll refer to both. I'll just read it from there and then I'll explain it to you. Okay, so what we talk about is stroma and lumen. Okay, stroma is chloroplast we're talking about. Okay, so stroma is all the outer side. This is the thylakoid membrane. Okay, this is the lumen, the inside of the stroma, you can say. Okay, and ATP synthetase is the enzyme. It is made up of you can say two subunits. One is F0 subunit, which is embedded in the membrane, thylakoid membrane. Okay, and this F1 portion, that is outside. Okay, H plus ions, they are there inside the lumen, and then you have photosystem 2, photosystem 1. If you remember, when we did the ETS chain, we had the photosystem 2 and photosystem 1 both. Okay, and as a result, we had at the end we had two energy bonds formed. We had ATP and NADPH. Okay, so NADPH is formed and then ATP is formed from here. So children, what it is, how it is done, thylakoid, so this much from here, and you very well know that ADP plus pi plus one phosphate molecule will give ATP. So this is the structure over here. I'll try and come back again to the blackboard. Now if you have your textbook, you can open it over there. So what it says is that there has to be a proton gradient across a membrane. Now what is proton gradient? If you remember, when we did splitting of water, in splitting of water we had hydrogen ions, that is protons. Okay, and over here when ATP synthesis has to take place, the proton gradient has to be there. What do you understand by gradient? Gradient is the difference. Okay, मतलब protons का जो number है वो अंदर और बाहर में difference होनी चाहिए. So we have greater accumulation of protons inside the lumen, okay, as compared to outside. And even whatever hydrogen ions are there outside also, they enter through this cytochromes, means electron transport chain mein. Okay, it is transported and finally it enters inside. And hydrogen ions, क्योंकि difference होता है, so again it passes off through this channel which is created by ATP synthetase. If you remember F0 F1 molecule, so through this F0 the H plus ions comes to the stroma. So actually this is the whole story about this chemiosmosis. I'll try and read out for you. Okay, so it says that like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. So this is a membrane, across a membrane, proton gradient is inside the lumen. Okay, it is more and outside it is less. So finally the movement from inside the lumen to outside that is in the stroma through the channel created by this F0, F1 and enzyme. Okay, so this is what takes place. So again, there is a proton gradient accumulation towards the inside of the membrane, that is in the lumen, I have told you. Okay, and in respiration, protons accumulate in the intermembrane space of the mitochondria. So mitochondria और इसमें रेस्पिरेशन और फोटोसिंथेसिस में यही डिफरेंस है बेटा. रेस्पिरेशन में इंटर मेम्ब्रेन के बीच में वहां पे एक्यूमुलेशन ऑफ प्रोटॉन्स होगा रेस्पिरेशन में, और ये फोटोसिंथेसिस में इट इज एक्यूमुलेटेड इन द ल्यूमेन. सो दैट इज द डिफरेंस. ओके. And the electron movements through ETS, yes, through electron transport system, that takes place. Now the proton gradient across the membrane, we need to consider this. So first, splitting of water takes place. तो जो हम लोगों ने पहले पढ़ा है in the first point, okay, on the inner side of the membrane. Inner side of the membrane मतलब splitting of water अंदर lumen में होता है. So these hydrogen ions or protons, whatever you say, that is released over here. Okay, the electrons, everything is released over there in the inside of the lumen. Okay, and the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. Lumen of the thylakoid ke lumen mein they get accumulated. And then as the electrons move through the photosystems, you know ki photosystem 2 P680, okay, it requires a wavelength of that to get excited, and then the electron movement takes place. Okay, it reaches to PS1 where P700 is the accepting wavelength. Okay, and finally it goes to the LHC light harvesting complexes है, फिर NADP NADPH है. Okay, so as the electrons passes from PS2 to PS1, okay, so hydrogen ions, through the hydrogen ions, that is what it says. Okay, so protons are transported across the membrane. So as electrons move through the photosystem, protons are transported across the membrane. So जैसे electrons move करेगा, okay, through the photo, electrons transport in this ETS chain which is there, PS2, PS1, cytochromes. Okay, so these are the transport systems, and finally these protons come out, that it crosses the membrane towards the outer side of the membrane, transfers its electron not to an electron carrier. So again, one very important thing, it says that the primary receptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier. Okay, so primary receptor of electron is located towards the outer side of the membrane. So finally, how will the electron move? So electron carrier is hydrogen ion. So hydrogen is the one which carries the electrons towards the outside. Okay, so electron moves through hydrogen. Now this molecule removes the proton from the stroma. So finally this stroma again, from the stroma one hydrogen ion is removed. Got it? The electrons move out, and then along with the hydrogen ions it moves to photosystem 2, and finally it comes again into the lumen. So one of the hydrogen ions from the stroma is removed, is moved. Okay, while transporting an electron. एक electron को transport करने के लिए one hydrogen ion from stroma again comes back into the lumen. Okay, when this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side of the lumen side. Okay, so this is how it takes place. The electrons which are there, it moves out and finally combines with the hydrogen carrier. The hydrogen carriers are there, and hydrogen carrier, through hydrogen carrier the electron is brought to photosystem 2 for ETS chain, and along with that hydrogen ion भी है, तो hydrogen ion comes to the lumen, increasing the concentration of hydrogen ions in the lumen. Got it? And second one, the NADP reductase enzyme is located on the stroma outside. Okay, NADP reductase enzyme, that is located in the stroma, and along with electrons that come from the acceptor of electrons of PS1, protons are necessary for reduction of NADP to NADPH. So protons are also important for the reduction of this NADP to NADPH. So it plays a very important role over there as well. Okay. Yes. So within the chloroplast, protons in the stroma decreases in number, while in lumen there is accumulation of protons. Hope you are able to understand this. And this is what creates a difference, a proton gradient, proton difference. Okay, across the thylakoid membrane, outside and inside there is a difference. And this is what actually, okay, the movement is what actually helps in releasing the proton gradient, breaking the proton gradient and release of energy. Okay, due to the movement of protons across the membrane to the stroma through the transmembrane channel. So how is this? You can say the gradient broken. That is movement of protons through the this channel. This channel. This channel is known as transmembrane channel. So through this, the hydrogen ions moves off, breaking the gradient and releasing energy. Energy is released. Okay, and F0 and F1, I told you this is the enzyme ATPase synthetase. This is made up of two units, F0 and F1. F0 is embedded in the thylakoid membrane, F1 is outside, and through it forms a transmembrane channel through which the hydrogen ions is removed, and finally this ADP, okay, gets oxidized to ATP. It forms ATP, and this is how actually the ATP energy bond, okay, is formed. ATPase, which makes the enzyme synthetase, okay, and several molecules. This is ATPase. It forms the enzyme ATPase synthetase, and finally the energy packed ATPase are formed. So children, the last paragraph, chemiosmosis, it requires a membrane. What all is important? It requires a membrane, which is thylakoid membrane, okay, a proton pump. This is a proton gradient, proton pump, okay, a proton gradient, proton pump, yeah, okay, then a proton gradient and ATPase energy. Okay, so ATPase enzyme, all these are the requirements of chemiosmosis. Okay, and energy normally is used to pump protons across a membrane. Across a membrane, again from lower concentration to higher concentration movement hoga, so energy is required. So energy is used over there to create a gradient or a high concentration of protons within the thylakoid lumen. Okay, and ATPase has a channel that allows diffusion of protons back across the membrane. So this releases enough of energy to activate ATPase enzyme. So the energy which is released, okay, in breaking the gradient, the energy which is released, that activates this ATPase enzyme, forming ATPase synthetase, which helps in the forming of energy bonds, that is ATP. Okay, and along with the NADPH produced by the movement of electrons, the ATP is also used immediately in the biosynthetic reaction, that is in the photosynthetic reaction, okay, in the stroma, which takes place in the stroma, which is responsible for photosynthesis, fixing of carbon dioxide and synthesis of sugars. So this is the whole story about ATP synthesis through chemiosmosis. I hope even when you read your book, you will be able to understand. Okay, so go through line to line. I have tried and explained it to you from here as well as through your textbook. Any doubts, you can always ask me. I'm not going to go beyond that. Okay, there are other topics, but we'll take it in the next class. Thank you all. Thank you for watching. Do keep watching. And as I told you before, many of you have not subscribed. Children, one class, like you can always go and see the videos yourself. Okay, so you'll have to subscribe it for that, because many important questions, many important things will be coming on the channel every now and then. So you will get the notification, and besides that you have quite many things, book reviews, motivations, quite many things. So do subscribe the channel, share it with your friends and like it. Thank you all. Thank you for watching. Thank you so much.